mean, I think it is a spectrum.
Yeah.
You know,
True.
I think there definitely is a spectrum.
I just never saw the severe one.
I saw one that was so severe, so, I don't know how the parents. That is a cause of divorce, I would say.
And there's so
The
many,
parents
like,
get so stressed.
so many things to think of, you know, like if you see someone who was normal to this age and then became autistic, how do you work that out? And so much to think about. Like I said, my mom is just, oh God, I don't have to do that stress anymore.
Yeah, so
Make sure you're used to the DSM-4 criteria. Doctor, likes DSM.
Everything's already set up. So
Okay, so how about everybody else? Is
How do you
any,
work
They'll
with
have you
Delegate?
printed
Who
out
else is here
from, like,
that's
the first
coming?
.com website.
I mean, actually, for
I
the
like him
basic
so much.
science, everybody's
Yeah,
supposed to be
somebody-
here.
I'm like, oh, Mariana. And then he started talking to me in Spanish.
I told him that, like, I took it in college, and the rest of every visit was entirely in Spanish.
I mean, where's Joyon? Where's Lakshmi? Where's, I
Lakshmi was fine.
mean,
Yeah, actually.
Lakshmi is there, and we're gonna check.
Okay.
taking a French fry. Fantastic. Oh my God, that's nice.
And when I come to tournament.
Annie's on his way.
Yeah, Annie.
Yeah.
I mean, everybody's
Yeah,
busy,
already.
but neuroscience, Anusha, good.
You're like it's an ACLS right now.
I think I know.
Lakshmi is coming.
No, she's just doing her, like, she wants to do her.
Very, very flat.
Yeah, it was,
Which I like.
I love it.
Why, what's about the Friday?
Here?
No, there.
Anusha, are you all set for this evening?
Very, very.
Good. Where are you going? Estia? Okay, good.
Yeah.
I don't know, I just book whatever they do.
And you're prepared to introduce Dr. Berger,
Yeah. I have to
Okay.
find
or will
Dr.
be
Farmer.
prepared? Yeah.
I thought that she was going to, but I'll.
Okay, why don't you, as long as somebody is ready and knows that they're gonna do it, or I'll do it if you want me to, but let me know either way.
He said that yesterday. I love it. You have one of the
Yeah, well, when I was there, there were 4 people. There was Jefferson and there was two other people. Not enough room in there. We were just like,
What does everybody say? So, Lakshmi and John Young. Who else? We need some, so Dilip is away. Is he?
He's on EEG, but he's
Dilip.
said he has ACL. John Young should be. I know he was seeing a patient earlier. So.
And hi, Paul. Yeah. No. Okay. Good. Okay. Okay. I would, yeah, that, why don't you give her a gram? How big is she? How big is she? Oh, then just give her 750 of Kaplan. Okay. Yeah, I think, okay. And where is she gonna be, in PACU? Okay, good. I'll let the team. Okay, okay, thanks. So there's going to be an urgent consult at 1 o'clock in PACU on the patient who just had a second stage AVM repair and had a seizure coming out from anesthesia. So anesthesia gave her some bolus of propofol. They'll give her some Keppra right now. So need to be seen as soon as we're done. I know everybody's tongue is hanging out right now. It is busy. I get it.
No, I actually it was ICU.
In PACU. She's came out of, so she's still anesthesia's patient and will stay anesthesia's patient until we deal with her.
Another consult?
Consult, not necessarily neuro ICU. So I mean, it's consult. So one more, Lakshmi. Okay, where's Lakshmi?
Lakshmi, I think she's getting.
His wife?
She's getting her lunch right now.
Okay. Tell her to ask what her ETA is. I'm going away for a week, so right after Grand Rounds tomorrow, I'll just share with you that I am aware of how busy it is right now. I'm also aware of how disruptive the patient flow is because of the fact that basically we have 6 neuro step-down beds in the hospital that are shared by us, neurosurgery, IR, and spine surgery. So I'm attempting to make management aware of this. They're pretty aware of it because the whole hospital is yelling, I can't get neuro step-down patients out of my unit, wherever their unit happens to be. So I'm aware of that. And I'm trying to share that with people who have the resources to do something about it. So I can come, okay?
Which patient are you talking about?
I don't know the name of the patient. She was the young girl from, I think, the Dominican Republic who she's already had a stage 1 AVM repair. This is new. I don't think they were anticipating we'd be involved in this. Good. Hi, Lakshmi. Okay, let's focus on basic neuroscience. For those, welcome to all the Jeff students. This is each noon conference, each day of the week is devoted to a different big topic. Thursdays are basic neuroscience. And today, this year is neurophysiology, and today is about calcium signaling. Okay. This is also the introductory lecture to this bigger topic, which is second messengers. I think all the rest of the second messengers will be talked about next year in neurochemistry, neuropharmacology. Calcium is the only one that spills into this year. So let's just talk about what a second messenger is, all right? A second messenger is a molecule that relays a signal from the extracellular space to the intracellular space, okay? That's what all second messengers have in common. So they are not the cell surface receptor, and they are not the implementation of the intracellular signaling transduction. They communicate between the receptor on the surface, okay, and the intracellular mechanism. And this intracellular mechanism depends on the presence of that second messenger, and the second messenger is generated by an interaction with the cell surface receptor or channel, okay? So that's what all second messengers have in common. And then we're going to look at calcium as a second messenger, okay? We're going to look at calcium as an ion itself, and then some calcium physiology, and we're going to spend a lot of time on calcium flux. And this is a huge topic. I mean this is a very important topic, okay? Because this is virtually ubiquitous throughout the body, and especially on all excitable tissues are going to depend on some mechanism involving calcium. I think that it helps to look at the size of an ion, because you're going to see, so I want you to get a sense of charge density. I often think, like, what's the difference between sodium, all right, and calcium? Look at this. The sodium ion and the calcium ion are the same size. Okay, so how does the body, how does a channel, or how does a receptor, or how does a protein know the difference if they're the same size and they're both cations? So it has to do with this idea of charge density, okay? This calcium, which is the same size as the sodium, the one electron floating, okay, in an external cell on a big atom, a big nucleus like the sodium, that electron can float away. It doesn't take very much. But to remove two, okay, that means that this ion has a significant concentration of positive charge, if you will. It has lost two negative charges, and this thing is exerting a field which is substantial around itself, okay? And you look at little magnesium, okay? This thing is very concentrated, more concentrated than the calcium is concentrated, okay? And magnesium is smaller than lithium, okay? So it's worth, and look at the size of the potassium. I mean, the potassium, it lose an electron from a thing this big, and all these electrons floating around on 3 orbitals filled, and it wants to lose 1 electron. I'm like, who cares, you know? Sodium, only two shells, and it loses 1. Lithium is only 1, okay? But these guys are divalent, okay? And you don't see anything like this in the body. So the +3, you're not going to see. As a result of which, that charge density is going to attract not only counter ions, chloride, bicarb, that sort of thing, but it's also going to interact in electrostatic interactions with the water molecule itself, okay? And the water molecule, oxygen, will orient themselves toward, because this is carrying a 2-electron lobe on it. If you were to look at the electron orbital, there's 2 electrons that are paired, so they don't go wandering away, okay? But they're there. There's a significant negative charge density on this side of the oxygen opposite the hydrogen. So they orient themselves to the calcium, and the calcium almost never is going to exist naked. It's always going to have a water shell. And if a protein wants to interact with the calcium, it's going to have to displace a water molecule, okay? Like this. So now you see and the calcium, because it has this big charge density, it can change the conformation of a protein. When it comes in contact with the protein and the protein goes, oh, I like that, like an aspartate, right? See, no accident that we've got aspartates and glutamates, because they're negatively charged amino acids, okay? So now you've got these negatively charged amino acids, and you throw into it this incredibly dense charge of positive, okay? not surprisingly, you get this electrostatic interaction. And this is a very powerful force, which is strong enough to change the conformation of the protein. Okay, that's the basic. That's why calcium can function as a second messenger, because look at this. These are actually the same apolipoprotein that is out there. This is a ApoS100 protein, and just look at this little piece of it. Look at where the green is, and look at how the yellow is oriented. Now throw a calcium into the mix. This guy stands up. The yellow helix is dragged with it, and the green comes flipping up over, okay, to interact with it. Just a simple example of some major conformational changes that occur when calcium binds to a protein, okay? So it will open pores, expose sites which can interact with modulatory proteins, that sort of thing. This is so the calcium is a very, very powerful agent. Now, having said that, the way that the body acts is to have extremely low levels of cytoplasmic calcium. So we're talking 10 to the -7. That's a 1/10 of a micromole of calcium floating around in the cytoplasm, which is not much. Okay, that's an extremely low level. Okay, but when you activate the cell at the peak, it's going to hit millimolar or 2 millimolar concentration. So it's going to jump 4 orders of magnitude. So the calcium in the cytoplasm is going to rise 4 orders of magnitude. This is a significant signal amplification. This is a high signal to noise ratio. You're going from nothing to the top of the mountain in one excitation. Okay, so what we need to see is, how does this happen, and then how does the body get rid of all of this calcium fast enough to be excitable again, at 100 hertz, okay, 100 times a second. So how is it doing this? All right, that's what we want to see. All right. The consequences of this are that this mechanism, this calcium influx mechanism, is used for nearly all excitation contraction in both cardiac muscle and skeletal muscle and smooth muscle. It's used in signaling for neurotransmitter release. That most receptors are going to interact with this. Not all, but most. And even if they don't interact with it immediately to let the calcium in, they're going to interact with they're going to modulate a calcium-oriented system in most cases. And then we're also going to say that if you really flood the system with calcium and you don't eliminate it quickly, then you're going to give rise to long-term changes in the cell. It's going to take that as a prolonged high concentration of calcium is going to be taken as a signal to the cell that either it needs to commit seppuku, okay, and kill itself, okay, or it needs to learn to deal with this, and that's what learning is about. These high sustained calcium levels actually are very often, especially in the nervous system, the signal that you need to learn. You need to change the structure of your dendrite. You need to change the structure of your synaptic terminals. You need to throw more receptors, or you need to increase your capacity to handle this cell, because this is going on all the time. And that actually is the foundation of learning, okay? All right, so let's go to the voltage-gated calcium channels, okay, which is actually 3 families that we're going to have to deal with. So there isn't a single, like when we send clinically and we say, we'll send off a voltage-gated calcium channel, okay, there are about 200 of them, okay, but they all are based on a few subunits. So let's look at this, all right. First of all, nearly all excitable cells have voltage-gated calcium channels. I don't care what part of it, what organ we're talking about. If the cell is excitable, there's probably going to be some voltage-gated calcium channel associated with the excitation. What the structure will change. You're going to be able to modulate these basic peptides that make the voltage-gated calcium channel. And in particular, for instance, from experience, that people who are excited, right, if they're on edge, right, when they go to jump, okay, you jump higher, right? So how does that happen? Well, the adrenaline surge, right? Right, that's correct, okay? Well, how does the adrenaline cause you to jump higher? And the answer is, well, it modulates the voltage-gated calcium channel of the skeletal muscle. That's the answer. Okay, so we need to see all that. We need to see that all of that laid out. Okay. Here's the basic. This is just a cartoon, and it's a generic cartoon. So here's your basic voltage gated calcium channel pore, and it's pretty closely related to the sodium channel. It's you know, evolutionarily, these two are real close cousins, and they diverged a very long time ago and not very far from one another. And then you're going to have these modulatory proteins. This is extracellular. You're going to have an alpha 2 and a delta. This is not a sympathetic alpha. This is just its name. And then you have a gamma subunit and a beta subunit. And the point is that once the calcium comes in, it may be used for contraction, it may be used for secretion, it may be used for learning and modification of the nucleus. All of those can occur. This is an old slide that is still useful because it's historically, we still use this terminology because our pharmacology is still driven very much by this. So the first type of calcium channel was found was an L type, and the L stands for large, okay? And what you have, look at this, okay? These are three characteristics. For the L type, okay, you have a high activation voltage. So this is -10. You're on your way, right? You're thinking sodium potential, right? This is a regenerative potential. This cell's depolarized. If it's a neuron, okay, it's firing, okay? It has a large calcium conductance. It's going to flood the cell. It's just going to open the gates and calcium's coming in, high volume of it. And it's very slow to inactivate. So this doesn't sound like a good neuron mechanism to me, because neurons don't work this way. You flood the cell and you take your time about getting rid of it. That doesn't sound much like a neuron to me, okay? At the opposite extreme are the T-type cells, which have a very low voltage of activation. We're basically at the resting membrane potential here. And they have a little sip and then spit the thing out like that. That's what we're doing for the T-type. Okay, so a small conductance that's inactivated very fast. Believe it or not, that doesn't quite sound like a good neuronal stimulation either. And these tend to be your oscillators. These are going to be your pacemaker cells. Are going to use a T-type. Why? So that they can oscillate. So the thing is, the kinetics are so fast that the cell can oscillate if it wants. That's what a pacemaker does, is oscillate. Okay. These are your neuronal types, NPQR, okay, which are intermediate between the two. Intermediate voltage activation. You don't want to do this at resting membrane potential. You want to move away from, you know, you're depolarizing the cell. It lets in enough calcium for the neuron to know it, but it's able to get rid of it pretty quickly if it wants. That's the neuron type. All right, let's look at the structure of the voltage-gated calcium channel. Alpha 1 subunit is the pore. So that's your basic thing. And all you need to do is have an alpha 1, throw it in a membrane, and it'll let calcium in. Okay, but there's no controlling it. It's not useful or physiologic. It just lets the calcium in, and that's not going to be very good. But that's your channel, is right there, and it's darn similar to the sodium channel. The beta subunit, which was extracellular, no, sorry, the beta subunit was the intracellular unit, is an alpha helix and totally intracellular, and that will impart voltage sensitivity to this channel. Okay. You don't need to know which is which. I don't think it matters. But that these exist is what I want you to know. The gamma is a glycoprotein. That's convenient. Gamma being a glycoprotein. All right. And that has to do with putting the voltage-gated calcium channel where you want it. If you remember, we talked about being able to move proteins in the membrane bilayer plane and then assemble them into groups. So the gamma subunit is the one that is important for that. The alpha 2 subunit also has voltage regulation, and the delta, okay, anchors it into the membrane. So if we go back, that what I didn't point out, so look at the delta. Now that has a little lipid tail. Remember that was one of the mechanisms by which we anchored a protein into the membrane, was to grab a lipid tail. And there's a glycophosphatidylenosyl inositol, a glyco sugar, phosphatidylenosyl. So phosphate group, inositol, head group, okay? And it's the same thing that we had before, same cartoon. Here's your pore, Here's, this is for trafficking. This one imparts voltage sensitivity. This is your real voltage sensitivity. And this guy anchors at orientation in the membrane. This is another cartoon which just shows you the chains, if you will. There's not new information up here, but here now is what's important, and that is that we don't call them by these letters anymore. They're in these families where the L type is called CAV1, so voltage gated calcium channel family 1, that's L, okay? Voltage gated calcium channel type 3, family 3 is the T type, and the neuronal type, the intermediate type, is CAV2. So if you see a CAV1, you figure it's going to be in a muscle cell of some sort. If you see a CAV3, it's going to be a pacemaker. If you see a CAV2, it's probably going to be in the neuron. Okay? Like that. And I don't want to go over it, but notice all the CAV2s are neurotransmitter. All the CAV3s are pacemaker. All the CAV1s are excitation secretion, excitation contraction. Okay? So this works. We don't need to go over the detail there. All right. Now, let's watch this dance of calcium flooding the cell and leaving the cell. All right. So first of all, we said we start with extremely low calcium. All right. Secondly, then we're going to have either a ligand-gated neurotransmitter, right, or voltage-gated depolarization, opening of channels, all right, and the voltage-gated calcium channel, which is activated either by ligand or voltage. So you could have also ligand-gated calcium channels. They let calcium in across the plasma membrane. So all of your ionotropic receptors do that. Glutamate, acetylcholine, GABA. If it's an ionotropic receptor, it's going to interact with calcium across the plasma membrane. And so is the P2X, not P2Y12 group, not P2Y, but the P2X group of ATP receptors are calcium channels. They're direct calcium channels. All right. There's not as much, even with the L type, if you will, which we don't use in neurons, but even that doesn't really let in that much calcium. We need to amplify the signal, right? And we amplify the signal by having these huge calcium stores in endoplasmic reticulum, which get released. That's the bulk of the calcium that's going to hit the cytoplasm. So we might, through extracellular action, we may take the calcium level from 10 to the -7th to 10 to the -5th. That's a big jump, but that's nowhere near 10 to the -3rd. I mean, these are orders of magnitude larger, okay? So those two big orders of magnitude are actually going to be an intracellular release, okay? Not extracellular flooding the cell. So here's the amplification. So you have this voltage-dependent stimulation of intracellular release, meaning that the voltage-gated calcium channels, which are actually L-type, sitting in the endoplasmic reticulum or smooth reticulum, are going to activate a ryanodine receptor. And the ryanodine receptor is going to have the calcium leave the endoplasmic reticulum to enter the cytoplasm. I'll show you pictures. Now, what about all these metabotropic receptors? Now, by definition, a metabotropic receptor does not directly change any ion gradient, any ion conductances, by definition. But these metabotropic receptors, meaning the metabotropic glutamate, acetylcholine, ATP, all of these, ATP through the P2Y12, all of these modify the receptors at the cell surface so that they then, and this will be next year, we're going to talk about activation of phospholipase C, which is going to generate diacylglycerol and it's going to generate IP3, all right? And those are what's going to open up the endoplasmic reticulum. So we're not going to be talking about that except indirectly today. So here's a cartoon, and we're only, if you're red, you're on the activation side. So we want to focus on red for the moment, not blue. So calcium is let in by a receptor or a voltage-gated calcium channel, either one, and the calcium comes in, and then it activates a series of second messengers. Now these which open up the ryanodine receptor, and this is what floods the cell with calcium. Got it? All right.
It's easier to make up just because the ryanodine receptor is also involved in myasthenia.
Mhm.
So now we are seeing it backwards. So it's
Oh yeah, and
easier to-
dantrium acts at the ryanodine receptor. Okay, so yeah, we deal with this pharmacology all the time. Okay, all right. At any rate, once the cell gets to the point, all right, that it is depolarized to about 10 to the minus fifth, it starts to think about closing, and so you start closing these channels as they're releasing this flood of stuff. Now, if you hit 10 to the minus third calcium in the cytoplasm, and it starts interacting with, let's say, a phosphate group, it's going to precipitate. And pretty soon, you're going to have chalk in the middle of the cell. Okay? So you can't do that. Okay? You don't want a cytoskeleton. You don't want a shell, and you don't want a crustacean inside your neuron or your muscle. So you're going to have to protect the calcium from all of these divalent negative ions like phosphate, okay, and HCO, HCO2 really. So what you do is you buffer it. So you need to buffer your calcium, all right, when it's at these high levels. And you have two basic sources of buffering. The first is you have a level of buffering that involves organelles, and the main buffer is going to be the mitochondrion. So we don't talk about the endoplasmic reticulum as the buffer because that's the storage site. So we don't want to talk about that. But the mitochondria is capable of absorbing massive amounts of calcium. All right, we'll look at that in a minute. It's built to do that. And then you have hundreds of calcium binding proteins. Now, I don't want to leave you with the sense that these hundreds of calcium binding proteins are there just to soak up calcium. Okay, that's part of your signal transduction mechanism. The calcium is going to bind to these calcium binding proteins, and there's a lot of them, and they're all over the place, and they will bind a lot of calcium, but they're not doing it just for buffering purposes. They serve a buffering purpose, but that's not why they're there. They are signaling, okay? So, all right. And the other thing the cell can do, if you remember back to our lecture on synapse, we spent a couple slides on the gap junction synapses. So another way to get rid of calcium is to give it to the next neighbor and just let it through the gap junction to go into an astrocyte, okay? Or put it to another cell. So you can buffer via other cells, you can buffer via your mitochondria, and you buffer via your proteins. The mitochondria actually has a calcium. This is the mitochondrial calcium uniporter. It just lets the calcium in. It's a uniporter. There's no exchange. You don't have to do anything. Calcium concentration goes up. The mitochondria says, let it come in. Okay. If it's a little bit increase, it lets a little bit in. If it's a lot increase, it lets a lot in. Now, it'll get to the point. So the mitochondria is I always think of it like a horse eating oats. A horse will eat oats until they make themselves sick and fall down sick. Okay, they won't stop. There's no satiety for a horse eating oats. And the mitochondria is the same way. It will let calcium in until it kills itself. Okay, so the pathologic, one of the signals of apoptosis is a release of cytochrome oxidase from the mitochondria. That release is occurring because of the calcium overload of the mitochondria. So it can't really protect itself. And it's going to be passively injured by massive overload concentrations of calcium. It's a uniporter. There's no real regulation of the system. And this was a slide from our gap junction talk. All right, so now you've let this calcium in, you've released it from the extracellular source, you've buffered it, however you're going to buffer it, now you want to get rid of it. So here's the situation. I used to have to lyophilize phosphatidylcholine in the lab. Okay, in order to lyophilize something, you have your chemical that you want in a solvent. It's in a big beaker. Okay, and you need to do two things. You need to get rid of the air real fast so it doesn't make a gel, okay? And then you really need to suck hard and get rid of everything that you possibly can. One pump cannot do that. You can make a pump which will take a lot of volume quickly, where you can make a pump that will suck hard, but you can't make a pump that will suck a lot of air and then pump hard. The cell has to have the same problem. It needs to quickly eliminate a lot of calcium, and then it needs to drop it down to 10 to the minus 7th. And it can't do that by a single mechanism. So you have a high capacitance system with low affinity, and then you have a low capacitance system with very low affinities. Okay? So the way that you get, and you have one for extrusion and one for intracellular uptake. So the extrusion mechanism is high capacity, high volume, all right, but low affinity, is the sodium calcium exchanger. So I'll show you a picture, but it just, you have the sodium gradient already set up, lots of extracellular sodium. You just take that and let the sodium in and throw the calcium out. And that's your sodium calcium exchanger, okay? And that will get rid of most of your calcium very quickly, but it will not get you down to a low level of calcium that you need to be at basal. So then you turn on an ATPase, a calcium-specific ATPase, and you spend the energy. Now, that won't be a high-volume system, but you can suck as hard as you want on the calcium and get rid of every last bit of calcium. And you're going to have similar system on the intracellular side, where you have the smooth endoplasmic reticulum exchanger, okay, that will bring it back up into the endoplasmic reticulum and then finish the job with a plasma membrane calcium activated cell. And then one last problem. You just let out a lot of calcium into the extracellular space with the sodium exchanger. Your cell is now, your cellular stores are now depleted of calcium. So it left the endoplasmic reticulum, it went into the cytoplasm, and then you just threw it into the some of it, not all of it, but some of it went into the extracellular space. You do that a few times, you're going to run out of calcium in your stores. So then you have to have a way of replenishing the store, which we do. Obviously we have to, so we do. All right, so cartoon time. All right, so this is actually a T-tubule. This is, which we'll come to in a minute, but this is an excitable cell. We have on the striated muscle, we have an L-type calcium channel in the T-tubule. It lets the calcium in. The calcium comes in, all right, and is modified, by the way. The effect of the calcium on the ryanodine receptor is modified according to the presence of cyclic AMP, okay, or G proteins, all right? And so then the amount of calcium which is released from the endoplasmic restore is modified by the presence of the norepi, all right? You do your thing, you activate your, okay, then you want to end the system. And so you have the uptake system and back into the endoplasmic reticulum. You do it with a low capacity system, a high capacity system. You exchange your calcium via the sodium exchanger. Okay. Of course, you have to replenish that, which you do with an ATPase. Okay. Here's how you refill the store. There's actually a channel here in the endoplasmic reticulum, which is activated when the calcium level inside the store starts to fall. And it goes to the membrane. So it actually drags itself up to the plasma membrane and it says, oh, feed me, mama. Okay. And it interacts with a calcium, with a voltage gated calcium channel, and it just opens it. It says, I need calcium. So you have a specific protein in the endoplasmic reticulum that grabs hold of a calcium channel and just opens it. And it says, just let the calcium come flooding in. I need it. I'll let you know when I'm done. And it just sucks on the extracellular calcium and brings it in directly. You don't want it in the cytoplasm. That would be an activation of the cell. So it goes directly from the extracellular space directly into the endoplasmic reticulum. I mean, these are cool molecular mechanisms. I think we've basically talked this. And this is just a nice cartoon. This is, you don't need, the cool thing about muscle is you actually don't have to have calcium enter the cell from the extracellular space in muscle. All you have to do is let the calcium come in via the T-tubule, and these will directly open voltage gated calcium channels directly educate the endoplasmic reticulum to release its calcium, which it then does, which activates the actin-myosin system. So that's nice, very direct system. Now, if you, however, if you want to do it via the neuromuscular junction, so that's a different matter. And this is again, this is the calcium in the T-tubule, which is let in directly activating the endoplasmic reticulum without having to let calcium into the cytoplasm. But it will. It's going to flood the system in a second. As soon as it knows it, it's just going to release the calcium. Which also means that it doesn't need to extrude a lot of the calcium in the muscle cell. It's going to put it back. All right. These are via CAV1 families, and we've already talked about the catecholamines stimulating it. So, and any questions? I keep talking, but it's, yeah, are you following what we're saying? So it's a cool, and this is the same picture. So we've seen this. All right. Excitation, secretion coupling, which is neurotransmitter release. Okay, it's pretty simple because we've already had the synaptic, we've already had this discussion. And you remember that there were specific proteins that would interact with the snare proteins that imparted calcium sensitivity to them. So all we need to do is get the calcium in, which it's pretty simple now, given the background that we have. We need CAV2 channels, all right, to let the calcium in via either a voltage-gated calcium channel or directly via a ligand-gated channel. And then that stimulation will release more from the extracellular stores and will interact with the mechanism that we know that will drag the synaptic vesicle to the surface, okay, and then cause it to fuse. And we can modulate this system with G proteins, with adenylate cyclase and that sort of thing. So we modulate the whole system, but you get it. There's nothing really new. It's the same as what we've already known, okay? All the principles that we just talked about, calcium, are the same, and we've already done this. And so this is done by calcium. We got it. And this cartoon is just here to show you the modification by the G proteins or the modification by the cyclic nucleotides. Okay? So all this, and modified by other things. Here you have the calcium being taken up into an astrocyte, right? And activating nitric oxide synthetase. Okay, and you have calcium stores in the presynaptic terminus. Everything like we've talked about. And then T channels are our pacemaker cells. So you see them at the sinoatrial node. You could see them in right. Who's the frequency generator of the central nervous system? Is the thalamus. So it's in the thalamus that you're going to see these, in addition to all their other activity. We're going to spend a minute on this. In addition to all the normal activities of activation by receptors and that sort of thing, that we're also going to have to deal with pacemaker cells in the thalamus, the oscillators. And so we'll look at that. And the regulation is going to be via G protein cyclic nucleotide. So I found this slide last time I gave this talk, and I thought this was really helpful because I wouldn't want you to think that the question is looked at from the point of view of the electrical system. Don't think, let's not molecular for the moment, but let's think voltage. Okay, right, just voltage. How much calcium are we letting in comparison to the sodium? Okay, so because this seems like, okay, voltage gated calcium channel, okay, this must be generating, does it contribute to the action potential? I mean, is it depolarizing the cell? No, the answer is no. This is a very specific mechanism via this high charge density, okay? This shows you the relative electrical conductance, okay, in amps, okay, picoamps, of the sodium versus the calcium, okay? So sodium is big, okay? The amount of electrical conductance is huge for sodium compared to calcium, okay? That's all that you get off this. This is an interesting thing because this is bursting. The idea is, how do you get bursting neurons? So bursting neurons can occur pathologically, but certain neurons, especially in the thalamus, burst on purpose, okay? So the question is, how do you get bursting neurons? And the answer with a CAV3 channel. So look at this. This is a knockout mouse with a CAV3 channel. It has no CAV3 channels in it, okay? And we are in the thalamus, all right? Now, if you wanted to polarize the cell, it behaves in the normal manner, okay? There's nothing wrong with its CAV2 channels. So whether you use an AMPA receptor or whether you depolarize it with an electrode or whatever you want to do, it responds. The wild type and the knockout behave the same way. But you want to stimulate out low, right? Low, close to the resting membrane potential. You want to stimulate bursting. That's easy to do in the wild type, but you can't do it in the knockout. So it's the CAV3 channel, voltage gated calcium. This is a CAV3, a voltage gated calcium channel, family 3, is involved in the bursting neurons in the thalamus. Now, these are knockout mouse, but we encounter this clinically all the time in pediatric clinic, okay? Because, I'm missing a slide, sorry. Okay, absence seizures, the inherited absence, classical absence, is a mutated CAV3 channel. And what happens is that the thalamus starts to oscillate at 3 per second because of the mutation. And that's the foundation of absence seizures. Okay? All right. A lot of stuff, but we're getting close to the end. I just want to talk for a minute about ligand-gated calcium channels. We've been talking about, all this has been on voltage-gated calcium channel. We have ligand-gated channels, and these are primarily the glutamate channels. So the acetylcholine receptor and the ATP, P2X, and that sort of thing is true. But the main receptor, the main ligand activation channel in the brain is the AMPA receptor, not the NMDA receptor, by the way. It's the AMPA receptor. And the AMPA receptor is like this guy, okay? It's very bland. It's just going to, on depolarization or by interaction with the glutamate, it's going to start firing, cause the cell to start firing. It's going to depolarize the cell. And the stronger you stimulate it, the longer you stimulate it, the more action potentials you're going to generate in the postsynaptic neuron. And that's it. It's just this continuous gradient. There's no threshold to it. You depolarize it, the cell starts. You depolarize it more, the cell does more. That's your AMPA receptor. And that's the large bulk of the system. AMPA receptors can be sodium specific, depending on the cell type, or may let calcium in. And that just has to do with the GLUT2 receptor. Okay, and they vary. They're going to be modified from cell to cell. That's the AMPA receptor. It's just very simple. You just turn it on, crank it up, roll it down, crank it up, turn it down. The NMDA receptor is less prevalent, less frequently used, but much more complex. And what's going to happen is that if you activate an NMDA receptor, you're going to have a long-term persistent calcium activation in the cell, okay? And that's going to alter the long-term performance of the cell. It's going to learn, right? So you better not do this very frequently because you don't want to be mislearning the wrong stuff, okay? So you don't do this all the time. So therefore, it's not just glutamate. You're going to have to depolarize the cell. You're going to have to unblock the magnesium. You're going to have to activate it in addition with a second key with either serine or glycine. So the NMDA receptor is very protective. You can't just do this only with glutamate and activation by the presynaptic neuron. And when you do it, it's going to result in long-term changes in the cell. It's going to change its dendrite structure. It's going to change via a CREB mechanism. It's going to activate genes within the cell that are going to produce long-term structural changes in the cell. That's your NMDA channel. And you sort of know that, okay? And then the metabotropic receptors we've already talked about. They modify this whole system, okay, via things like phospholipase C and other things that now modify the ability to release. So it's just a constant modification system that eventually get down to the gene level. all right? And these are the problems, not the problem, these are the functions that are taken care of by the cell. The number of synapses, the dendritic complexity, the response to growth factors, the long-term synaptic strength, and memory, all are depending on this NMDA type activation, all right? But this is a different level than the AMPA receptor activation. And again, these are the things that, this is 2 or 3 lectures next year. This slide constitutes 2 or 3 lectures next year. All right. All of them activating via a calmodulin-dependent calmodulin kinases, and that's something that I do not want to get into today. Okay. And all the way to gene transcript. Okay. So this is just a summary of what we've seen. And we've already said it. I think we've had enough. Just one more thing. These oscillations are interesting. How do you decode oscillations? How do you know if you're oscillating fast or slow? This is an interesting question. Okay. So the calcium calmodulin kinase, I'm going to show you a slide that's going to answer it. But the answer is that the oscillation rates are going to be integrated via a calcium calmodulin kinase mechanism. Look at this. So watch this. The calcium calmodulin kinase is tuned, okay, so that it will ignore just independent calcium, okay? But if you start putting a bunch of calcium fluxes together, so its tuning is such, its kinetics is such, that it starts to accumulate activity. And this is what's happening. So if you low frequency calcium oscillations just keep occurring, and you return to baseline, and you return to baseline, and you return to baseline. But if you pick up the speed of the oscillation within the domain, the time domain of the kinase, then the signal starts to build up, and it doesn't return to baseline. That's what's happening. This is your mechanism for sensing oscillation frequency. All right, and post-atonic potentiation is also occurs via a flood of calcium, typically through the NMDA receptor, sustained long-term calcium. Remember also, I'll be done in a second, Paul, but you remember that the peptide release, remember the small molecule neurotransmitter release occurred with a single activation. But to activate the peptide, it wasn't even releasing the peptides into the synaptic cleft. It released them at other parts of the synaptic terminal. And we said that required major stimulation. Okay, this is the mechanism for the release of the peptides from the big dense granules. And this is the basis of post-atonic potentiation, which I assume you're going to have a lecture on from Chris Bradley or Jan later in the year. But this slide is, and then we said, if the calcium really goes up, then the cell commits seppuku, okay, and just kills itself, and that's apoptosis, and this is the basis of apoptosis. And these were there's a slide I was looking at. Here's the mutated CAF3 receptor in an epileptic child with absence. Okay, so calcium is a big event.
Get your.
Yep. Hey, Paul, how are you? Come on in. I could probably answer messages from now until the end of the day. What came in this hour. I would love something.
Yeah.
Hey, Paul.
Hey, so this overall, it's still seizing. Keppra, CT's negative. The company says DSMO does not potentiate seizures, but I just looked it up and it can. So
I've seen some of those.
That's the solvent for
Oh, yeah.
the
Right.
products.
Yep.
So
Does she seize even if she's on